Okay, so we're going to pick up where we left off from last class. We left off talking about fibroblasts being the predominant cell type within connective tissue, and the predominant cell type that's also that secretes most of the ECM molecules, particularly within our connective tissue ECM. To take a moment to try and describe another component of our connective tissue ECM are the glycosaminoglycans, also dubbed as GAGs. They're these disaccharide or two sugar repeating units, and they're typically distinguished because there are different species of GAGs, distinguished by what kind of sugars are present, the kind of linkages between them, where whether the linkage might be between specific carbon groups, and the number or location of sulfate groups on those GAGs. The four main types are hyaluronan, chondroitin sulfate, and dermatan sulfate, heparin sulfate, and keratin sulfate. What's important to note is that as the name might imply, the keratin sulfate GAGs are ones that are typically found within our hair and within our nails. Most notably, chondroitin sulfate is an example of one where one that's in the brain, and that typically scar formation that occurs within the brain is really rich in chondroitin sulfate. So the main takeaway here is what GAGs are, that they're these repeating disaccharide units composed of amino sugars and uronic acid, that there's most of them are sulfated. Hyaluronan is the one that is not sulfated, and we'll talk about more about hyaluronan a little bit later, but it's important for you to try and understand here, like, that largely GAGs are these repeating disaccharide units and how they can be distinguished from one another. So typically GAGs, they have these large extended conformations. They want to take up as much space as possible. They, relative to their mass, they have a high volume to mass ratio, meaning they want to take up as much space. So if you imagine riding the bus into grounds, and like the really annoying person that takes up a lot of seats, that like spreads their legs or arms and is taking up an obnoxious amount of space, that would be a GAG within the connective tissue ECM. They have a high volume to mass ratio, and because of that, they are able to form these really porous structures within these collagen networks that are in our connective tissue ECM here. What's really key for their functional roles here is that they have a negative charge that attracts cations here, and that negative charge makes them want to repel and stretch out from their own from other GAGs that are around them. So that's part of the reason that there's this repulsion that makes them want to take up a lot of space. Oftentimes, there's also a high osmotic pressure where water will flood in and help it retain its structure, help it maintain the swelling pressure. And this is important in helping our tissues that are rich in GAGs, that are able to withstand high compressive forces. And so when there's this compressive force coming in that is basically contracting GAGs together within our connective tissue ECM, it's gonna push water out, and there's gonna be this negative charge that's gonna want to repel them away from each other, but also a high osmotic pressure that wants to push water back in. So again, there's this repulsion of the negative charges, as well as an osmotic pressure that wants to drive water back in when these GAGs are pushed together and wanting to allow water to flow back in. Well, we're going to describe that a little bit more in the context of tissue to help make that a little bit more sense. Here is a table. I'm not going to ask you to memorize everything on this table, but just to help try and point out some of the unique features of the different GAGs that we have here. One that is not one that I do want to draw your attention to particularly is heparin. That's the GAG with the highest negative charge density, and it typically is known for its role as serving as a depot or reservoir for growth factors or proteins to bind to. So some of the parts and pieces of our connective tissue ECM or extracellular matrix, they don't just provide they don't just provide a physical role, they don't just provide a biological role to cue cells what to do, but they'll also actually hold proteins or growth factors in the ECM that can be really important for when they're enzymatically released to like activate cells to do certain features. And so there's this really cool and I think compelling idea. This is me starting to get into the weeds of science again. There's this idea of like growth factors and proteins being stored in our ECM as a way of like the matrix keeping a memory of stuff, where if there's a really inflammatory event, there can be inflammatory proteins secreted that are like held into the matrix that helps the matrix remember. And it's a way for consecutive inflammatory events to elicit more inflammation over time, where it just holds there and is ready to be released to activate cells again. So key take-home message here is that different GAG species have different unique features. One that I want to point your attention to here is heparin in particular has a high negative charge relative to others, and that it can actually serve as like a depot or a reservoir for growth factors. We talked about this a little bit last class, but just to draw your attention to it again, we're going to talk about proteoglycans. And again, to distinguish between proteoglycans and glycoproteins is if you look at the suffix, that'll tell you what that molecule is mostly comprised of. So proteoglycans are mostly composed of sugars or carbohydrates by about 95%, whereas glycoproteins can typically range from about only 1 to 60% of carbohydrates. So it's typically, on average, more protein than sugar or carbohydrate. Proteoglycans typically have a core protein, and that typically has GAGs that are added on. One thing to note is that the addition of sugar molecules to a protein, we call that glycosylation. That's the process of adding sugars to a protein. And that typically occurs within the Golgi apparatus, and it exits the cell via exocytosis. Typically, proteoglycans, depending on the degree of glycosylation, the degree of how many sugars are added or how many GAGs are added, helps them vary in size. So decarin, and we have these two proteins, these two proteoglycans, forgive me, illustrated below here. We have decarin, which has a core protein with 1 GAG chain on it, whereas we have aggrecan, which has a core protein, which I believe is, well, forgive me, has a core protein with hundreds of GAG chains coming off it, where leading to decarin having a molecular weight about 40 kilodaltons and aggrecan 3 megadaltons. And you can see a electron micrograph image below that helps to illustrate what a proteoglycan looks like biologically, as well as another illustration of aggrecan here. And to note, typically the species of GAGs that can, the species of GAGs are not always, each GAG that's on there aren't exactly all the same. It can be different species of GAGs. Yes?
Is like the amount of GAGs proteoglycan has that measured by percentage of molecular weight, or like, so when you say like proteoglycan can contain 95% carbohydrates, that like 95% of their molecular weight is in carbohydrates?
Yes. So some of the key functions to take note of for proteoglycans here. They are actually really key for the assembly and organization of our ECM. For an example, with decarin, it actually has a collagen binding region, and so that enables it to help contribute to collagen organization within our connective tissue ECM. It can actually regulate the traffic of molecules in cells, and so we talked about how heparin has this growth factor binding domain, and so it can actually regulate the availability of molecules. It can typically cells have receptors that enable them to bind to the GAGs or sometimes the core protein on these proteoglycans, and the porous network that they create also helps to regulate their ability to migrate through and traffic through. When we talked about their compressive strength, the particularly misspelled it there, not aggrecan in cartilage tissue. And to try and illustrate what that looks like here, we have a collagen fibril all around here. We have the hyaluronin core protein there, and then we have a hyaluronin molecule there, and we have aggrecan coming off the hyaluronin. So what ends up happening in our cartilage tissue here, we have compression. So let me take a step back, forgive me. So without compression, at rest, for those of you sitting in chairs right now, in your knees that don't have much compression in them right now, water is, there's an osmotic pressure that drives water into our connective tissue ECM here in our cartilage tissue that helps create this shock absorber or dampening effect that keeps our cartilage healthy. And so what ends up happening is when there's a compressive force, it pushes out that water, and these negatively charged gags are forced into close proximity with one another. What ends up happening now is the tensile strength of collagen helps create some strength and rigidity here. But in particular, the two key things to take note here for resisting compressive forces is the repulsion from these negatively charged gags in close proximity, as well as a osmotic pressure that wants to drive water back in. And so that helps, those two key driving forces help to provide resistance or provide compressive strength to our tissues rich in gags, rich in proteoglycans. I'm going to stop and give space for a couple of questions here. Yes. The repulsion of the negative charge of the gags wanting to functionally push back and create space from one another again. Because again, they want to have a high volume to mass ratio. They want to take up space as in an effort to try and stay away from these adjacent negatively charged molecules. Yeah, so the osmotic pressure of water is pushed out of this small region here under compressive pressure, but there's going to be a driving osmotic force, osmotic pressure that wants to drive water back in. And so those two things together help provide compressive strength in proteoglycan rich tissues. And so, for example, as for those of you who are runners, running is typically a high impact sport or activity. As you age, you start to lose some of those proteoglycans, you start to lose some of those gags. And so what some runners over 40 or over 50 will do is they'll actually take glycosaminoglycans or chondroitin sulfate pills to try and replenish the proteoglycans and gags in their knees so that their cartilage tissue continues to remain healthy. Thankfully, I think most of you don't have to worry about that quite yet. Yeah, so I will keep, yes.
Kind of half answer that. How does this relate to the cartilage loss, like or something?
Yeah, so that's a good question. So so there, our cartilage typically, cartilage can serve two functions. So we have like our cartilage in our knees that withstand compressive forces, and we have cartilage on articulating surfaces that basically like move around. So we have like, that would be our hip, that would be our shoulder, elbow as well, and there's some degree as well with our knee, but you can think of, let's stick with the knee. So we have an articulating surface in the knee where you don't want bone on bone rubbing against each other, but you also need to be able to withstand those resistive forces, those compressive forces. And so your meniscus in particular helps to withstand those compressive forces, but then there's cartilage that helps to provide an articulating surface to make sure that we don't have bone on bone rubbing against each other. They serve two different functions. Articulating surfaces don't need to withstand very much compression, but our meniscus does. And so there's going to be different ratios of proteoglycans or GAGs in those different kinds of cartilage. And so, and also if you think about this isn't something we've talked about yet, but our ECM is able to remodel in response to load and forces. And so, for example, like if you think of this is an example when I took Physio 1 when I was here, but there's like the example of like the tennis elbow. Like our bone remodels based on how much load and how much use it is. And so like if you look at I think it's like the radius and the ulna of a tennis player, or maybe it's the humerus. But there's remodeling in response to forces. And so the same goes with our ECM as well. It remodels in response to forces. And our cartilage, based on where it is, as well as under what kinds of loads it's facing, will remodel to meet that demand. There are people that have degenerative diseases where at 40 years old, they need a hip replacement. And then there are people who just over time, that articulating surface is just worn away over time. And so that's why after 7 years old, like there's a dramatic increase in people who need a hip replacement just because it's just worn away over time. These are great questions. Yes. Yes. All right. So within our connective tissue ECM, part of what constitutes that, in addition to proteoglycans, is also going to be fibrillar proteins. So we're going to take a moment to describe some of those because they're key. The key function that fibrillar proteins provide is to resist tension. And so one that does that is collagen. So collagen is the most abundant protein in mammals. Bless you, for those of you that have ever made bone broth before, where you just kind of take a chicken carcass and boil it down and you get this like gelatinous, quite delicious broth. If this is a bit morbid, if there was a bone broth made out of us, we would be, that broth would be basically 25% collagen. And so it is the most abundant protein in mammals. We talked before how it has 3 alpha chains. It has this triple helical structure. And so to try and make sure that we're not confusing concepts here, we talked about, secondary structure. We have like the alpha helix. This is collagen. We're talking about collagen protein here. It has 3 alpha chains together that form a triple helical structure. It's not anything to do with the secondary structure of alpha helices. So it's easy. This has been like one sticking point, like one area of like things can kind of overlap and be confusing. And so this is just saying that collagen is formed in 3 alpha chains that form this like helix together. And we talked before that, so that's one key feature of collagen. And it's made with these Gly-XY repeats. X's and Y's are usually proline and hydroxyproline. We'll talk a little bit on the next slide about what hydroxyproline is and why that's important. But a key word that I want to underline here is that that this is usually proline and hydroxyproline. It is not exclusively proline and hydroxyproline. That's a key thing to note here. And as we talked in the previous lecture, that collagen is formed when it's synthesized intracellularly in its pro-protein, pro-collagen form, its precursor immature form. And so there have been 42 different alpha chains identified, which have led to 28 different isoforms of collagen being identified and described. Yes?
Real quick, so is each alpha chain considered a pro-protein, or?
That's a good question. So it's the entire triple helix of collagen together that's considered to be in its pro-protein form. There's different alpha chains, but there's different genes that associate different genes for each alpha chain. For example, whenever we want to target cells that express collagen, an easy one that people typically do is we'll target collagen 1 alpha 1. And so like there's just different alpha chains available, but when we talk about the protein, that's the triple helix together. So for the GlyXY repeats, the glycine is really important. For those of you that have memorized every amino acid and know its structure, its abbreviations, its molecular weight, you'll remember that glycine has the simplest side chain of all. It's this really small amino acid relative to others, and it is benign, and so it's hydrophobic. And typically things that are hydrophobic want to be buried and basically like shoved on the interior of a protein. And so what you'll note here is, I'm not going to fathom what color that is, forgive me, but on the XY repeat on the left here, there's a smaller, not letter labeled ball. That's the glycine that serves as the backbone for collagen. And so typically it's on the interior of the alpha helix. The X amino acid, the X motif, is typically proline. And again, that proline is really important because it kinks and provides that turning that helps form the alpha helix. The Y is typically hydroxyproline. And so that hydroxyproline is pretty important because it is hydroxyproline is hydroxylated proline, and it is unique to collagen. So there's no other protein that has hydroxyproline in it. And that's really unique because if we wanted to look at how much collagen was in a big chunk of tissue, we could totally disassociate and make totally degrade down, like if we took a whole lung and degraded it down, and if we want to see how much collagen was in there, we could do an assay where we just measure how much hydroxyproline is in there and use that as a surrogate to compare against other tissues to see how much collagen is there. And that's one way we sometimes look at scar formation in organ tissue to see how much scar formation has occurred. We use hydroxyproline content as a surrogate to do so because that typically denotes how much collagen content is available. Yes.
So, you said there's 14 different alpha. Is that like different? Like some might have the proline, hydroxyproline, some might have like different concentrations of this. What is the difference?
So it's down to the amino acid sequence. Like some of them will have different different amounts of proline and hydroxyproline. But we'll actually, if you hold that thought, there's going to be a table. Oh, I forgot. It's the next slide after what I was going to look at. So we have a bunch of different collagen types that have different physical forms. So collagens 1, 2, 3, 5, and 11 are all very fibril. And so those are going to have not exact same sequences, but pretty similar. Then there's going to be the network forming sheet-like collagen, that's type 4, that forms a part that is a part of the basement membrane. And so that's going to have far less proline in it. It's still a GlyXY repeat because that is a defining feature of collagen, but it's going to have less proline and hydroxyproline, or it's going to have less proline and hydroxyproline in it relative to other types. There's also more recently defined collagen 27 and 28 that are non-fibrillar, and 127 being a transmembrane one. And if you can imagine, we talked earlier in the semester about a hydropathy plot where we look at, as we move across the amino acid sequence, we can kind of look at how hydrophobic the region of that protein is, and that'll tell us that was used to define and determine that collagen 27 is a transmembrane protein because there was at least 25 consecutive hydrophobic amino acids. And it was also probably imaged and defined to be visually confirmed to be in the transmembrane. Yes?
Are all three chains the same to each other, or do you have two different alpha chains in the same polymer?
So that's a very good question. So there are typically different alpha chains within one collagen. And typically the different combinations will denote like a different isoform of collagen. If that makes sense. So the key take home here, I'm not asking you to memorize everything on the slide. And as a general rule of thumb, if there's a table with a lot of information, I'm not going to ask you to memorize that. But what I do want you to take home here is that there are many different kinds of collagen, each of them with a different function, each of them with a different tissue distribution. Typically, what degree of tension resistance they provide or tension strength they provide will kind of inform where they are in the body. And also if there is a defect with that specific form of collagen, each of them has some sort of downstream consequence of that. Again, not to belabor the point again, not asking to memorize them, but just understand that there's many different kinds of collagen with different functions and tissue distribution sites. So collagen is present in there's a structural hierarchy to it. So we have our single alpha chain, and we know that a single collagen molecule has a triple helix of those 3 alpha chains together. Now, what will end up happening is we have a pro cap on both ends of collagen. It'll be enzymatically cleaved. And so now we have mature collagen that can now assemble together. And the way they'll assemble is collagen in its fibrous form is different than collagen type 4 for the basement membrane. If you imagine kind of like a little kid's train track where you have the train cars that like link together end to end, that's how these collagen molecules will assemble together. They go end to end once their pro caps have been cleaved. And once as that happens, there's also a lateral association. So let me use my mouse here to try and indicate that. So we'll have multiple collagen molecules linking together end to end, but then they'll begin to laterally associate into these bundles that we call collagen fibrils. And these fibrils are typically about 10 to 300 nanometers in thickness. And then from there, those fibrils will laterally associate together to form a collagen fiber, which is on the order of several microns. And so cells, in particular fibroblasts, their approximate diameter is about 30 microns. And so when they're moving across and grabbing onto our ECM, they're doing that at a collagen fiber level. But again, it has a hierarchy where we go from the single alpha chain to a triple helix pro collagen that's cleaved and they link together end to end. You get lateral association of these fibrils that are also then again aggregating together to form collagen fibers. And so once we have our collagen fibers, the way in which these fibers are oriented within our tissue are actually really important and dependent on what tissue it is. So one example here is tadpole skin, which is similar to has a similar orientation to our bone. Collagen fibers are actually oriented. We have these sheets that are oriented perpendicularly to each other, one after the other. Another example is tendon. If you think about your bicep tendon, that tendon moves pretty uniaxially. It's just moving along one single axis. And so all of those collagen fibers are oriented in one direction because if you think about the function of what that tendon is doing, it's moving along one axis. So everything is aligned in that direction. So the key take-home message here is that the tissue-specific organization of our collagen fibrils and fibers is dependent on what is that tissue doing, what loads is that tissue undergoing. So we have these collagen fibers, and if they're moving and providing resistance to tension, it's important to consider how are they stabilized in response to all that tension. One way in which they do that, one way in which that occurs is the formation of crosslinks here. So there's a key enzyme that's important for that formation of those crosslinkings. It's called Lysol oxidase. So again, enzymes can sometimes have big scary names. if we break it down piece by piece, we can decipher what Lysol oxidase does. So oxidase, it is catalyzing an oxidation reaction. And the Lysol part is telling us that it's catalyzing an oxidation reaction on lysines, okay? And so it is also a, and so what's occurring here is that it's catalyzing the formation of aldehydes on these lysines, on collagen. And these, one thing to note is that these aldehydes are highly reactive, meaning that when these aldehydes will undergo spontaneous reactions with other Lysol oxidase derived aldehydes or even unmodified lysines to form a cross-link across them. And so what ends up happening is that this creates a stability across our collagen network within our connective tissue ECM. Are there any, I will pause for a couple of questions. Are there any questions here? Either from this slide or the last few slides? Yes. A key role that these crosslinks are providing is that they're creating, these crosslinks provide a stabilization of our collagen network within our connective tissue ECM. Yes.
So the collagen fibers get cross-linked to other collagen fibers?
Yes, and that actually is a way for us to see how mature collagen is. So if it's been like newly formed, newly synthesized collagen, they won't quite have crosslinks yet. And so that's typically an indication of like mature aged collagen networks. Yes. Yes. Next piece of our connective tissue ECM that we're going to take note of is elastin. It's a highly hydrophobic protein in our ECM. Similar to collagen, it has a common repeat that defines, that is characteristic of this protein, the VPGXG. Unlike collagen, unlike other proteins, there's no glycosylation on elastin, nor any hydroxylysine, but there are hydroxyprolines that are present here. When it's made in its soluble precursor form, that's tropoelastin, and it's typically secreted out into the extracellular space. One key, and the crosslink, there's similar cross-linkings that happen within elastin that's similar to collagen. So what we have here is to the top right, there's an electron micrograph of a section of an aorta. So elastin helps provide, after collagen provides strength in response to some sort of tension event, elastin helps tissues return back to their structure. If we think about our skin is, pretty strong, and the collagen helps provide it and provide strength. But for our skin to be able to return back to its original shape, that's the role that elastin provides. Here, what we have is a section of the aorta, which again, if you imagine the aorta is right outside the left ventricle and has to endure huge bursts of pulsatile flow, and it needs to be able to return back to its shape. And so these large vessels are elastin rich. And if zoomed in on this region of the aorta, you can see there are cross-linked, there are elastin fibers rich there. Two key pieces or two key qualities of these elastin fibers, that they're incredibly hydrophobic, and that's again key for its ability to recoil back to for tissues to recoil back to their original shape. And there's also these alanine and lysine rich alpha helical segments that are key for the cross-linking that can occur here. Forgive me, apparently I need to sign back in. I hope that's okay. All right, we'll press on. If I get booted off, we'll fix that. There's an organization or a hierarchy to these elastic fibers that we need to take note of for a moment. So elastic fibers have an elastin core. This gray region here on this elastic fiber is an elastin core, and there's these fibrillin glycoproteins that bind that are on the outside and that bind to elastin. Again, elastin is very hydrophobic, so it's going to want to be buried on the inside of these elastic fibers. So what ends up happening is these elastic fibers are kind of coiled together in tissue in a relaxed state. When it undergoes stretch, it exposes these elastin molecules that are highly hydrophobic, typically in this aqueous environment that the tissue that comprises of the tissue. And once that stretching force, that tension force is released or relaxed, when there's enough elastin, it'll enable the recoil or return of that tissue to its original state or its original configuration. And so the key take-home message here is that it's the hydrophobic properties or qualities of elastin that cause it to be buried within an elastic fiber, and that when it undergoes stretch, that enables it to return back to bury itself when it's typically in an aqueous environment. The last connective tissue ECM protein that we're going to discuss is fibronectin. Fibronectin, interestingly enough, is a really interesting protein because it doesn't really provide much, if any at all, physical stabilization or physical strength to the connective tissue ECM. It's rather considered to be a signaling scaffold protein within our connective tissue ECM. And there's a lot of different domains present on fibronectin that make it this really bioactive molecule as opposed to a providing or serving some sort of structural or physical role in the connective tissue ECM. And so if we look across the entire segment of a single fibronectin molecule, there's actually like a few different types of collagen that's present, or not collagen, fibronectin present. So the rectangular regions moving from the N terminus down to the C terminus, we call that fibronectin type 1. The octagonal shapes we call fibronectin type 2, and the oval shapes we call fibronectin type 3. And so moving along from the N terminus down to the C terminus, what we can see is that there's regions that bind to heparin. And again, heparin is a growth factor depot, a growth factor binding gag that can bind to fibronectin. There's a collagen binding region. And then there's this really unique region that can bind to a wide variety of different integrand, a wide variety of integrants. We're going to talk more about what integrants are on the next slide, but we've already described them as receptors for the ECM. And so fibronectin is uniquely able to bind to far more than any other connective tissue ECM protein. And again, there's more binding domains further below on a fibronectin molecule. It is uniquely able to promote migration, differentiation, proliferation, can bind to a wide host of integrin receptors, and can also bind to other integrin, can also bind to other ECM proteins itself. And so again, it doesn't provide any mechanical integrity for our connective tissue, but it's uniquely able to promote signaling and binding to a plethora of molecules that other connective tissue ECM proteins cannot. So we're going to take a moment to discuss integrins. And so as we've described to this point, integrins are the receptors that our cells have for the ECM. And so it connects the cell to its matrix. It connects the cell to its environment. And there are 24 different kinds of integrins. They each have an alpha subunit and a beta subunit depicted here in what looks like green and blue. And there's 18 there's 18 different kind of alpha subunits and 8 different kind of beta subunits. And so the different combinations that occur in humans leads to 24 different heterodimer combinations of integrin. There's a long extracellular domain that engages with the outside environment. And then there's a short tail on the inside that binds to on the beta chain, on the beta subunit, forgive me, a wide variety of proteins that then helps anchor it to our actin cytoskeleton. And so what's really key here is that by engaging the actin cytoskeleton through integrins, cells can actually perceive the mechanical properties of their environment and exert forces on their environment. And it also enables signaling in both directions from the inside out and the outside in. These integrins typically, they have these different conformations that are allosterically regulated. When it binds and attaches, when an integrin binds and attaches to our ECM, it'll actually open up and like physically go from like bent to like a tall conformation that enables the intracellular domain now to engage with the proteins that target them and engage with the actin cytoskeleton. But also interestingly, these proteins on the inside can also trigger that activation. And we typically call that outside in or inside out activation. So the outside in, when it's typically in an inactive conformation, as shown to the left here, where the integrin molecules are bent, they'll unfold when it binds to the extracellular domain, and that will make the intracellular portion of the beta chain available and exposed to bind to the proteins that they are tasked with binding to, like talin and vinculin, as we showed on the previous slide. And then that'll lead to downstream activation where they engage the actin cytoskeleton and trigger a host of events downstream. But there's also inside out. So when it's in this bent inactive conformation, all these proteins that can bind to that beta subunit, they're competing to get there with and competing with the alpha chain or alpha subunit to get there. And so once that binding occurs, that can actually trigger the opening and unfolding of the integrin receptor and facilitate more easy engagement with the extracellular environment. And so key take-home message here, that there can be outside in activation where it's a receptor, so it binds to its ligand, our ECM, and drives activation downstream to the inside of the cell, or there can be inside out. The activation can begin on the inside of the cell and drive interactions outside or activation to bind things on the outside of the cell. I'm going to pause and give space for a couple of questions here before we move on. Yes. Yeah, so main takeaway fibronectin is that it does not provide mechanical integrity for our ECM, but rather it's a really potent signaling protein within our ECM that can engage with proteoglycans, engage with glycoproteins, engage with integrins. It is able to elicit a lot of responses by engaging with nearby cells or nearby molecules. Yes. The inside out. Yeah, so actually, I think if I go back one slide, that'll help, or two slides. So there are a lot of proteins here that are part of this complex, but two key ones to think about are talin and vinculin. In particular, talin is one that directly binds to the beta subunit of integrin. So inside out activation is talin when our integrins are in a bent inactive conformation. Typically, I'm going to take a step back. When our integrins are in a bent inactive conformation, I feel like I'm trying to do this with my arms and it's not working well. Typically, what that looks like is if we have a cell membrane here, I'll just draw them as single lines. They're close together, and I'll say this is beta and this is alpha, and there's not much space here for talin to come in and bind. But if they're open, they're separated, and now there's more space for talin to come in and bind. Inside out activation, what that means is talin is competing to bind to the beta subunit. It's competing against the alpha subunit. And when it eventually does bind, that again, when things bind, when one molecule comes and binds to a protein, that typically carries with it a charge that leads to a conformational change. So in this case, when talin binds to the beta subunit, it elicits the conformational change that happens with integrins. They go from bent to unbent to open conformation. And so once that happens, these arms of the integrin receptors are able to bind to their ligand, bind to the ECM protein directly around. And so that is typically what inside out activation looks like. Does that make more sense now? Great. Are there any more questions? All right, we'll press on. So we've been talking about how all of these different ECM proteins that make up the environment, the connective tissue environment for our cells. But clearly, there's not just constant synthesis always occurring. There needs to be a regulation that oversees and that regulates this. And so one way in which this happens is by ECM degradation, and the way in which this happens is by proteolysis. And so that's the breakdown of proteins. And a key way in which that happens is through proteases. Proteases, so it's an ACE, so it's an enzyme. Proteases are enzymes that break down proteins. They promote proteolysis. They break down proteins either into smaller peptides or into smaller subunits. And two key species of proteases to take note of are matrix metalloproteases, MMPs, or serine proteases. The matrix metalloproteases, again, these MMPs, it's a protease. So it is an enzyme that promotes, that targets and breaks down proteins. The metallo part means it needs a metallic ion as a cofactor to make the enzyme active. Cofactors are like other molecules needed to make an enzyme active. That it's a molecule that usually carries a charge that'll then change the conformation of the enzyme to make the active site available. Here in this case, it's usually a metal ion. And it targets the matrix. So we also just call them MMPs. And to the table to the right, we have a series of species of different MMPs, as well as what they target in its abbreviation, as well as other names that they go by. We also have serine proteases that, as the name implies, target serines. And in this case, they usually target plasmin and thrombin. The reason why this is important in the ECM is if you think about a cut, I feel like I usually go as like a cut to the hand is like the default example here. But if you cut your hand, you're bleeding, you have clotting that occurs to stop the bleeding. But that blood clot, that fibrin clot, is like an early matrix, a provisional matrix that helps guide and be an early matrix for like new collagen, new fibronectin, the new mature matrix that's eventually going to form. But that provisional fibrin clot matrix needs to go away. And so these serine proteases that are key with the clotting cascade also help to regulate this early matrix. And so one other key thing that they do is that these proteases help expose cryptic sites on various proteins as well during the wound healing cascade. And so again, cryptic sites are sites that are hidden that when made available elicit a different response. So how are these enzymes regulated? They're typically usually secreted as in their precursor form. And so they need to be activated by some sort of substrate typically. Sometimes they'll require a cofactor that provides one form of regulation. Again, the matrix metalloproteases, the MMPs, usually require some sort of metallic ion. They can also be restricted to the cell surface. And so we talked about some enzymes being available on the cell surface that orients and cleaves the procap from collagen type 4 or from laminin. And so there are enzymes actually on our cell surfaces that help regulate not only the maturing of different proteins in our ECM, but can actually help promote the breakdown or the degradation, the proteolysis of different ECM proteins. And finally, there's also inhibitors that can be secreted that help regulate these proteases. And so one example is an inhibitor that inhibits MMPs. They're called tissue inhibitors of metalloproteases, or TIMPs. And serpins are what inhibit our serine proteases. So to try and bring this all the way back to tissue engineering and regenerative medicine, the ECM is typically we typically try to leverage it to instruct how tissue should be created and regenerate or promote tissue repair. One way in which we do that is we'll take tissue and we'll decellularize it, which means we'll chemically treat it to remove all of the cells so that you just have the ECM structure there. And so that's kind of what the first slide from the last lecture, where there was an ear growing on the back of a mouse. That was a decellularized construct that was implanted on the back of a mouse, and that the scaffold of just ECM proteins provided the structure for cells to grow in and create this tissue. One other option, as opposed to decellularizing the ECM and using that, so there's a lot of what I should say. It retains the physical information, but there are a lot of unknowns with that. Sometimes, as someone from an immunology background, typically there's a lot of potential epitopes or immunologically reactive pieces still remaining, even on decellularized constructs. And so it's a great resource in research. But when we think about how do we move this to the clinic, there's a lot of unknowns as it relates to how immunologically problematic that might be. Another alternative is engineering biomaterials, engineering structures that are meant to mimic our ECM. It can have some, while it's I think super interesting, there is still some drawbacks. It's hard to totally recapitulate or recapture the actual structure of our ECM down to like, it's on this, some of our fibers on our ECM is down to the order of like hundreds of nanometers. And so it can sometimes be hard. It's possible, but hard at large scale to create structures at that resolution. And it can be, we have a lot of control over the physical characteristics and molecular characteristics, but it can be hard to control what are the physiological outcomes when you implant a polymer into the back of a mouse, or eventually into a human. And so I am, for the sake of time, not going to go through the I provided a research example of what some folks do here. So I'm actually going to, I think, jump ahead. This was about cryptic sites and fibronectin. So one that I'll show is largely how integrins can actually dictate. So this is like a case study in how this works. not necessarily something you're going to be quizzed or tested on. So you can sit back, relax, drink some water, take a sip of coffee. The whole idea here is that to try and illustrate that integrins are actually able to determine and dictate the integrity of our blood vessels. And so we talked about how integrins have an alpha chain and a beta chain. What's really interesting here is if you inject a mouse with, go back please, with some like fluorescent probe, what ends up happening is if you totally delete genetically all of the beta-1 integrins in a mouse, when you inject something into the vasculature of these mice, you'll see that their blood vessels become super leaky. And then also same thing occurs within the brain as well. And so this indicated that beta-1 integrins are super important for the integrity of our blood vessels. And what was done next was to try and look at, can we pattern specific integrin binding motifs to regulate the integrity of our of the vasculature here. And so they're able to show that if you play with like different fibronectin peptides that bind to either beta-1 or another type of integrin, you can actually promote the outgrowth and branching of blood vessels to grow and to become stabilized. And this is in particular important for angiogenesis, the growth of new blood vessels. And so like this is something that like our bodies are inherently capable of doing, from existing capillaries for like brand new capillaries to sprout off and grow. And what's really interesting is that if you block some integrins, you'll actually see no angiogenesis occurring. And so these integrins are not just important for the engaging to our extracellular matrix, but it's actually important for these biological processes that are important for repair. Finally, if you take a nanocapsule and put a fragment on it that binds to specific integrins, what you're able to see is that specific integrin engagement can change the degree of blood vessel stability. You can see like the blood vessels down here that bind to alpha, where there's a fibronectin fragment that binds to alpha V beta 3 integrins. These blood vessels totally change. They become way more tortuous and seemingly a little less stable. And so the summary here is that you can play with you can create constructs, specific peptides that promote engagement of certain integrins as a therapy to regulate and control how blood vessels can grow to promote angiogenesis, which is super important for tissue repair and tissue regeneration. Well, there was a whole host of people that I put on here, and I think I must have included that on the other slide, but I will post a slide that shows a whole host of individuals who have either informed my work, who have either mentored me, or who do some of the most incredible work in the country in tissue engineering or regenerative medicine. I'll post that on later tonight. And with that ends our lecture for today. You can? Thank you. Okay.